You know, I've been practicing medicine for a long time, and in that time, I've had to sit across from patients, good people, frightened people, and deliver a cancer diagnosis. And what I can tell you is that the conversation I have with those patients today is fundamentally different from the conversation I would have had even 15 years ago. 15 years ago, a lot of those conversations were about damage control. They were about how much of the burn and poison, radiation, chemotherapy a patient could endure. The tumor was the enemy, and we went to war with everything we had. The patient was almost collateral to the battle. Today, Today I talk to my patients about strategy, about mobilizing, about what's already inside their bodies that's been fighting for them this whole time, and how we can make it fight even harder. So what I want to do today is take you through the 5 most transformative shifts in modern oncology. And I want to do it in plain language, the way I'd explain it sitting across from a patient in my office, or honestly, the way I'd explain it to a family member over dinner. Because this is information that every single person deserves to understand. We're going to talk about something I call the starve and defend strategy. And by the end of this, you'll understand not just what's happening at the cutting edge of cancer research, but what you can do right now, starting in your own kitchen, to tip the balance in your favor. Okay, I want to start with something that, when I first learned about it in medical school, genuinely changed the way I think about the human body. And I think it'll change the way you think about it too. Here's the question. Do you think you might have cancer cells in your body right now? I know, that sounds like a terrifying thing to ask, but stay with me here, because the honest scientific answer is probably yes. In fact, almost all of us likely develop microscopic clusters of abnormal cells throughout our lives. These little aberrant clusters are constantly forming. Our bodies are imperfect. Our cells divide billions of times, and sometimes, not often, but sometimes, those divisions go wrong. A mutation happens. A cluster of abnormal cells starts to form. And yet, most of us don't develop cancer. Why? The answer to that question is one of the most elegant pieces of human biology I know, and it comes down to something called angiogenesis. Angiogenesis is essentially the process by which your body grows new blood vessels. It's a critical process. You need it for wound healing, for development, for all kinds of normal functions. But here's the thing about tumor cells. They need blood vessels too. They need oxygen. They need nutrients. Without a dedicated blood supply of their own, a tumor cannot grow beyond a certain size. And that size? It's about 2 to 3 millimeters, roughly the size of the tip of a ballpoint pen. At that microscopic scale, lacking oxygen and nutrients, the tumor just sits there, dormant, harmless. Your body keeps it in check through its own natural anti-angiogenic defenses. It's like a fire that never gets enough oxygen to actually catch. It smolders, but it doesn't spread. The danger, the real danger, comes when a tumor manages to hijack those blood vessel systems. When it essentially figures out how to signal your body to start growing vessels toward it, feeding it, giving it everything it needs to expand. And when that happens, it doesn't happen gradually. It happens explosively. I want you to really picture this, because I think the numbers will shock you. In laboratory experiments, researchers have shown that if you allow blood vessels to reach and actually begin feeding a tiny microscopic cancer cluster, that cluster can grow 16,000 times its original size in just 2 weeks. 16,000 times in 2 weeks. That's the difference between a dormant speck that will never harm you and a clinically significant tumor that can spread and kill. That's the biological cliff edge we're talking about. So the question became, can we keep cancer on the harmless side of that line? Can we prevent that angiogenic switch from being thrown? Can we cut off the supply lines before the tumor can build its infrastructure? And the answer, which we proved clinically in 2004, is yes. That year, the FDA approved a drug called bevacizumab, also known by its brand name Avastin, for the treatment of colorectal cancer. It was a landmark moment because bevacizumab is an anti-angiogenic drug. It doesn't kill tumor cells directly in the traditional chemotherapy sense. Instead, it targets the blood vessel growth signals that a tumor sends out. It starves the tumor by cutting off its ability to build its own supply lines. That's what I mean when I talk about the starve side of starve and defend. We're not just nuking everything in sight. We are using the biology of the tumor against it. But here's the even more extraordinary part. Bevacizumab was important, but it reinforced something we were already beginning to understand: that our bodies are naturally anti-angiogenic. This system already exists inside you. Your body already has the mechanisms to keep those microscopic cancer clusters in check. The drug didn't invent a new capability. It reinforced one that was already there. Which raises a fascinating question. What happens when that natural defense is weakened? What happens when the lifestyle choices we make, the food we eat, the chronic inflammation we generate, what happens when all of that slowly degrades our body's ability to suppress angiogenesis? We'll come back to that. But first, let's talk about the second revolution. And honestly, this one might be even more exciting. Let me tell you about a patient population that not very long ago we essentially had no good options for. Stage 4 cancer, what patients sometimes call the endgame diagnosis. Metastatic disease, cancer that has spread. For decades, that diagnosis came with a grim statistical prognosis. We could do things to extend life. We could manage symptoms. But the language around stage 4 was always the language of management, not cure. And then immunotherapy arrived. And I mean arrived, like a thunderclap. To understand why immunotherapy is so revolutionary, you first have to understand one of cancer's most insidious tricks. Cancer cells are your own cells. They came from you. And because of that, they have evolved over generations of mutations and selection to be very good at hiding from your immune system. Your immune system is remarkable. It is patrolling your body constantly, identifying threats, tagging and destroying anything that doesn't belong. Think of it like a police force. Incredibly well-trained officers, on the beat 24 hours a day. But cancer found a workaround. Tumor cells learned to express certain molecular signals, specific proteins on their surface, that essentially act as a cloak. When an immune cell comes close to a tumor cell, it encounters these signals, and instead of attacking, it essentially stands down. It receives a message that says, "Nothing to see here. I'm normal. Move along." This is not a metaphor. This is a specific molecular interaction, and it involves what we call checkpoint proteins. The two you'll hear most about are called PD-1 and PD-L1. PD-L1 is expressed on the surface of tumor cells. PD-1 is the receptor on the immune cell that receives the stand-down signal. When PD-L1 binds to PD-1, the immune cell backs off. Now here's where it gets beautiful. Researchers figured out that if you could block that interaction, if you could develop a drug that sat in the PD-1 receptor and prevented the tumor's cloak signal from landing, then your immune cells would suddenly be able to see the tumor. The cloak comes off, and your own immune system, which is far more precise and far more powerful than any drug we could manufacture, goes to work. These drugs are called checkpoint inhibitors, and one of the most well-known is pembrolizumab, which you may have heard of by the brand name Keytruda. The results have been, and I don't use this word lightly, transformative. We have patients, real patients, not hypothetical ones, who came in with stage 4 diagnoses with metastatic disease, who underwent immunotherapy and watched their bodies mount such an effective response that their scans came back essentially clear. We are not just managing their illness. Their own immune systems essentially destroyed the cancer. We talk about reaching a stage 0 status. That concept, a stage 4 patient reaching stage 0, would have seemed fantastical to an oncologist 20 years ago. And yet, it is happening. Now, I want to be clear and honest with you, because that's what I'd want from my own doctor. Immunotherapy does not work for everyone. Not every cancer is cloaked in this way. The checkpoint proteins need to be present. And even when they are, the response rates vary. Some patients experience dramatic responses. Others see partial responses. Some don't respond at all. And that brings us to what I think is perhaps the most jaw-dropping scientific discovery to come out of oncology in the last decade. Because researchers started asking, why do some patients respond and others don't? If the drug is targeting the same molecular pathway, why is the outcome so different? The answer, when it came, was not what anyone expected. I want to tell you about a scientist named Dr. Laurence Zitvogel. She works at the Institut Gustave Roussy in France, which is one of the premier cancer research centers in the world. And several years ago, she and her team were trying to answer exactly that question. Why do some patients respond to immunotherapy and others don't? They looked at genetics. They looked at tumor characteristics. They looked at all the obvious variables. And then they found the smoking gun. It wasn't the patient's genome. It wasn't the tumor-specific molecular profile, at least not entirely. The decisive factor, the variable that correlated most strongly with whether a patient would respond to immunotherapy, was the composition of the patient's gut microbiome. Your gut microbiome, the trillions of bacteria living in your intestines. Now, I know what you might be thinking. We hear about the gut microbiome in relation to everything these days. Digestion, mood, immune function, skin health. It sometimes starts to sound like marketing language. I get it. The word microbiome gets thrown around in wellness circles so casually that it loses its gravity. But what Zitvogel's team found was not wellness marketing. This was hard clinical science with real oncological stakes. Her team identified a specific bacterium called Akkermansia muciniphila. And I'll give you a moment to appreciate that name, because you're going to be hearing it more and more, as a critical gatekeeper of immunotherapy response. Patients who had robust populations of Akkermansia muciniphila in their gut were responding well to checkpoint inhibitor therapy. Patients who lacked it were not. And then they did the experiment that proved it beyond doubt. They took Akkermansia from human responders, from the patients whose immunotherapy was working, and they transplanted it into non-responding animal subjects. Animals whose gut microbiome lacked this bacterium. Animals that had not been responding to the same immunotherapy drugs. And here's what happened. When they transplanted the human Akkermansia into those animals, the animals began to respond to immunotherapy. Boom, like flipping a switch. Think about what that means. Take a moment with that. A multi-million-dollar drug, the most sophisticated oncological weapon we have developed, was not reaching its potential because of the microbial environment in the gut. The drug was fine. The drug was doing its job at the molecular level. But the biological soil in which it was planted was not ready to receive it. And when you change the soil, the drug worked. This is not a small finding. This is a paradigm shift, because it means that cancer treatment is not just about the drug. It is about the complete biological ecosystem of the patient. It means that a patient's lifestyle, their diet, their gut health, their history of antibiotic use, all the things that shape the microbiome, may determine whether a cutting-edge immunotherapy saves their life or doesn't. Now, to be clear, we are still in the relatively early days of translating this science into clinical practice. Fecal microbiome transplants combined with immunotherapy are being actively studied in clinical trials right now. We don't yet have a standardized protocol for microbiome optimization before immunotherapy in most clinical settings. But what this research tells us, definitively, is that the gut is not just a digestive organ. It is an active participant in the immune war against cancer. It is, in a very real sense, a command center. And that leads us to the most immediately actionable piece of this entire conversation. Because while we wait for clinical protocols to catch up, there are things you can do right now to nurture your Akkermansia populations. And the most powerful of those things involves something you do 3 times a day: eating. I want to share a piece of clinical data with you that I think should be in every oncology waiting room in the world, not in a pamphlet, in large print, on the wall. There was research done specifically in melanoma patients who were undergoing immunotherapy. Melanoma was a useful study population because it's one of the cancers where checkpoint inhibitors have shown particularly strong effects, which gave researchers good statistical power to look at variables that influenced outcomes. And they found something extraordinary about dietary fiber. Here is what the data showed. For every 5 to 6 grams of dietary fiber that a melanoma patient consumed per day, that's roughly the amount of fiber in a single medium pear, or about half a cup of cooked lentils, or a small handful of mixed nuts, the risk of mortality during immunotherapy was reduced by 30%. 30% per 5 to 6 grams of fiber. I want to be precise here because precision matters. This is not a study showing that vaguely eating healthy is good for you. This is a dose-response relationship between a specific dietary component and clinical survival outcomes in cancer patients receiving active treatment. That is the language of pharmacology, not lifestyle advice. Fiber feeds bacteria. Specifically, certain types of fermentable dietary fiber are the preferred food source for bacteria like Akkermansia muciniphila. When you eat fiber, you are essentially feeding your defenders. You are creating an environment in your gut where the bacteria that prime your immune system can thrive. And when those bacteria thrive, they produce signaling molecules, short-chain fatty acids, and other compounds that communicate directly with your immune cells. They keep your immune system, in a sense, switched on and primed for action. They maintain the biological readiness of the very system that immunotherapy drugs are trying to activate. So, the fiber is not working despite the immunotherapy. It is working with the immunotherapy. It is amplifying the same molecular pathway that the drug is trying to exploit. Think about what this means for the concept of food as medicine. We are not talking about supplements here. We are not talking about some exotic compound available only in a clinical trial. We are talking about food, real, whole, affordable food that you can buy at any grocery store and cook at home. Now, and I want to be careful to say this clearly, I'm not suggesting that eating a pear is equivalent to receiving pembrolizumab. Cancer treatment is complex. Immunotherapy is a powerful intervention that requires careful medical management. Please do not hear me saying that dietary changes replace medical treatment. What I am saying is this: the distinction between medical treatment and what you eat is less sharp than most of us were taught. The idea that nutrition is somehow separate from, or less important than, what happens in the oncology clinic is a false division. The evidence does not support that division. Your oncologist has incredible tools available to them, and the food on your plate is one of the most powerful tools you have access to. All right, let's talk about the kitchen. Because if the gut microbiome is a command center, and Akkermansia is one of the key generals, then the food you eat is how you supply and reinforce that army. I think of this as your inner pharmacy. The idea is that your body already contains a vast, sophisticated pharmaceutical operation. Your gut bacteria are producing compounds. Your immune cells are being regulated. Your angiogenesis defense systems are either being strengthened or weakened. All of this is happening all the time in response to signals that include, significantly, what you put in your mouth. So what specifically promotes Akkermansia mucinifera? What can you actually buy and cook to nurture this critical bacterium? Let's start with a class of compounds called elagitannins. These are polyphenolic compounds, don't worry about the name, found in pomegranates, dried cranberries, and Concord grape juice. When you eat these foods, your gut bacteria metabolize the elagitannins into smaller compounds called urolithins. And urolithins have been shown in research to directly stimulate the growth of beneficial bacteria, including Akkermansia. Pomegranate seeds in your morning yogurt, a glass of pure Concord grape juice, a handful of dried cranberries in your oatmeal. These are not exotic or difficult additions to your diet. Next, polyphenols more broadly. These are the pigment compounds in colorful, whole, plant-based foods. Bell peppers, especially red and orange ones, blueberries, raspberries, blackberries, dark leafy greens like spinach and kale. The deeper and more vibrant the color of a plant food, generally speaking, the richer it is in polyphenols. These compounds feed a diverse microbiome and support the anti-inflammatory environment your defenders need to function optimally. There are also some more specific and perhaps surprising stimulants of Akkermansia populations that research has identified. Oats and whole grains. The beta-glucan fiber in oats is particularly effective at stimulating beneficial gut bacteria. Your morning oatmeal is doing more than you might think. Chili peppers and dried chili flakes. Capsaicin, the compound that gives peppers their heat, has been shown in several studies to promote Akkermansia growth. Sprinkle dried red pepper flakes on your eggs. Use chili in your cooking. And, this one always surprises people, Chinese black vinegar. Also called Qingqiang vinegar, this fermented condiment has demonstrated effects on gut microbiome composition in research settings. It is widely available in Asian grocery stores and used commonly in dressings, marinades, and dipping sauces. Now, the overarching principle here is one of diversity. The research on the microbiome consistently shows that a diverse microbiome, one containing many different species of bacteria in healthy populations, is a more resilient and more functional one. The way you build diversity is by eating a wide variety of whole plant foods, not just one superfood, not a supplement that promises to fix everything, a genuinely varied diet of colorful, fiber-rich whole foods. Think of it this way. If your microbiome is an army, you don't want an army composed entirely of one kind of soldier. You want infantry, artillery, medics, intelligence, a full coordinated force. Different plant foods train different kinds of soldiers. And none of this requires a prescription. None of it requires a clinical trial.
Don't forget to hit that subscribe button and join the journey.
So, let me bring all of this together, because I want to leave you with something concrete. We have been through 5 profound shifts in how we understand and fight cancer. We've talked about angiogenesis and the power of cutting off a tumor's supply lines. We've talked about how immunotherapy has transformed stage 4 outcomes by pulling the cloak off cancer cells and unleashing your own immune system. We've talked about Dr. Zitvogel's groundbreaking work showing that the gut microbiome is a decisive factor in whether those treatments work. We've talked about the clinical fiber data from melanoma research. And we've talked about the specific foods that can nurture the bacterial commanders of your immune response. The thread connecting all of it is this: you are not a passive recipient of care. You are a participant. You are the commander of a biological army that is sophisticated beyond anything we can manufacture in a lab. Modern medicine is developing increasingly elegant tools to amplify and direct that army. And you have the ability, every single day, to feed it, strengthen it, and keep it ready for battle. Now, and I say this as someone who has practiced clinical medicine for many years, I am not minimizing the complexity of cancer treatment. I am not suggesting that any of what I've shared today replaces the judgment of your oncologist or the drugs they prescribe. Cancer is serious. Expert medical care is irreplaceable. What I am saying is that the old divide, where medicine was something done to you in a clinic, and everything else in your life was irrelevant to your outcome, that divide is gone. The science has demolished it. So here are 3 things I want you to walk away with if you or someone you love is navigating a cancer diagnosis. First, ask your oncologist about the molecular markers of your specific tumor. Ask whether your cancer has been tested for MSI-H status. That stands for microsatellite instability high, and whether it expresses PD-1 or PD-L1. These markers determine whether you're a candidate for immunotherapy. You have the right to know this about your own tumor. Demand the pathology specifics. Second, ask whether anti-angiogenic treatment, drugs like bevacizumab, or other targeted therapies might be appropriate for your tumor type and stage. The starve the tumor strategy is well established and FDA approved across multiple cancer types. It's not experimental. Ask about it by name. Third, go to clinicaltrials.gov. It's a free public database run by the National Institutes of Health, and search for your specific cancer type combined with the terms immunotherapy or microbiome. The frontier of oncology research is happening in clinical trials right now, and some of those trials may match your specific molecular profile. Your oncologist may not mention every available trial. Do your own search. Advocate for access. And then, come home. Make some oatmeal with pomegranate seeds. Add some chili flakes to your dinner. Eat the whole pear, not the juice. Feed your defenders. We are living through a genuine revolution in how cancer is understood and treated. For the first time in history, we are learning to win, not just by attacking the tumor from the outside, but by empowering the body's own defenses from within. Starve and defend. Cut off the supply lines and unleash the soldiers. You are not a bystander. You are the battlefield, and the choices you make every day in your kitchen, in your conversations with your doctors, in the clinical trials you seek out, those choices matter more than we've ever been able to prove before. Thank you for being here today. If this was useful to you, please share it with someone you love. And if you have questions, or if you want to go deeper on any of the topics we covered today, drop them in the comments. I read all of them. Take care of yourself, and take care of your microbiome.